Buongiorno e benvenuti a questo incontro.
Good morning and welcome to this meeting, being live. We will focus on the characteristics making plants, animals and human beings unique. Here, we will focus on the most elementary, the simplest organisms, though mysterious, and as we've seen in recent months, organisms that can have a significant impact throughout the planet, as in viruses. Today with us we have Professor Enzo Tramontano, Professor of Microbiology at the University of Cagliari. He deals with virology and he deals with the development of antiviral drugs, interaction of viruses with the innate immune system, and it deals with the study of endogenous viruses in human beings. He is now participating in a European project which is developing a vaccine against the coronavirus. So, good morning, Professor. To start with you, what are viruses? How can they distinguish themselves from other living organisms? Good morning. Viruses are living entities with two phases, an intracellular phase and an extracellular phase. The extracellular phase is called the virion and enables the virus dissemination between different cells and between two organisms. The second phase is called intracellular, where the virus replicates and therefore it enters the host cell and exploits it in order to form a progeny. This is very important in that viruses need another cell in order to replicate because they can't have their own metabolism and therefore they have to exploit the cell metabolism from a synthetic point of view. Therefore, viruses are obligate parasites, and as far as we know, they are present in all human beings. Let's think about man. There are pathogenic viruses such as SARS-CoV-2, which unfortunately is invading us, and also non-pathogenic viruses. So what is the structure of a virus? Now, the structure of a virus is very small. The virus is very small in itself, ranging from 20 to 400 nanometers, usually. And they have a protein coat, which is called the capsid, where there's nucleic acid, which can be either a DNA or an RNA. Outside the protein coat, there can be sometimes a double lipid coat taken from the host cell, which is called envelope, from where there's proteins coming out interacting with the host cell. What is unique in these living organisms is that their genome can be different from the genome of the bacterial or human cell. As we all know, all the cells have as a genome the DNA. However, viruses have a large variety of nucleic acids. We have viruses with double-strand DNA, with one-strand DNA, and also, what is unique in the planet, there can also be an RNA genome. So there's viruses with a double-strand RNA genome or a one-strand RNA with a positive or negative polarity. But this is unique in viruses. A very peculiar case, which I want to mention because this is very important in terms of interaction with man, is retroviruses. Retroviruses are very specific viruses where the genome is an RNA, but the RNA is copied into a DNA to then be transcribed into RNA. So there's a whole variety which you can see in the slide of viral genomes, but usually the genome, especially the RNA virus, which I'll use as an example, is quite simple. They are quite small, especially those of the RNA virus, and they have essential elements for the virus to replicate, to create the structure of the virus and its proteins, for the formation of the envelope. And then there's also additional proteins which are important in terms of interaction with the host cell. In the slide, you can see the SARS-CoV-2 genome, which has 30,000 bases. To better understand how a virus works, let's see how it replicates and how it enters a host cell. Every virus, according to its genome, has its own strategy to replicate. For example, and this is SARS-CoV-2. Here you can see that on the outer surface of the virion there's proteins, the spike protein, which have a viral receptor interacting with the cell receptor. So there must be the interaction between two receptors recognizing each other, two proteins recognizing each other, and a virus cannot infect all cells, but can just infect the cells having receptor recognizing them. Not all cells can be infected by several viruses. So there must be mutual recognition between a viral protein and a cell protein. Then there's several mechanisms whereby the virus enters the cell and releases its genome. In this case, for example, this genome is released into the cytoplasm. This is an RNA genome with positive polarity, and so it copies like a cell messenger RNA. Therefore, the virus deceives the cell, making it believe that the genome is its own. So the cell recognizes it as if it was its own genome, and so the RNA genome creates some proteins which are coded. These proteins also contain proteases, so smaller proteins are formed, as well as an RNA replicase. It is an enzyme recognizing the RNA genome, which actually does not exist in the cell. Usually a cell does not have this type of enzyme. Therefore, it has to be carried by outside. So it recognizes the genome, which has been released into the cytoplasm. It copies it. Then many other RNA messengers are made, leading to the creation of proteins. And at the same time, a copy of the viral RNA is formed. many other RNA genomes are formed. So the genome is packaged into this virion, which is then released. And this way, the virus has deceived the whole cell and has created hundreds of copies of itself. If we think about a pathogenic virus, or even non-pathogenic virus, when a virus enters an organism, like man, for example, the organism has systems to understand, to realize where, if a virus has entered. The easiest system is the innate immune system. So the innate immune system is made up of different sentinels, which realize whether there are viral portions in the organism. And as you can see in this complex slide, there's many different ways. There's many different sentinels recognizing different portions of virus. But in the end, in the slide, you can see the different sentinels converge into phosphorylation cascades, producing proteins. In these cases, they are interferons, but they can also be of other type. And in turn, these proteins activate other phosphorylation cascades, leading to the production of over 300 genes, which are the first line of defense of the organism against the virus. And this line of defense is quite important, so much so that viruses have co-evolved defense systems against this immune system, as in they codify proteins, as you can see in the slide, blocking different aspects of the cascades, and they evade, as in they disguise against the immune system. The more effective the disguise, the more pathogenic the virus can be. So, a virus uses the cell to replicate, as we said, and there's a sort of interplay between the virus and the outer cell, and the host cell. In order to better understand how we can block the pathogenic virus, how can we do that? I mean, how can we block the virus? We can block the virus. There are therapies, there are drugs which can be developed and used for that purpose. There are some successful cases. For example, we have drugs against the hepatitis C virus, or we have developed drugs against the HIV, or also drugs against other viruses. And these drugs actually block key elements of the replication, of the virus replication. So, for example, if there is a drug blocking the entry of the virus, then the subsequent steps will be blocked. If it blocks the viral protease, then the virus cannot mature. So there's many different ways in which drugs can block the virus, and there are many therapeutic targets. Today, research is trying to identify some drugs, some selective drugs against COVID. The drug which is used today was discovered for Ebola, but is also working on the polymerase of the coronavirus. So we've seen what a virus is, how it replicates, how it can use a host cell, and how the organism can block this replication. So we have an organism which, on the one hand, is simpler than all other organisms that we know, but is unable to replicate on its own. What is its role in the evolutionary chain? What is the origin of viruses? This is a very interesting question, but it's very difficult to provide one single answer to this question. Now, let's start from this image, which shows the universal phylogenetic tree, which is made by bacteria, archaea, and eukaryotes. As far as we know, in the tree, you see that there's many different branches in the three domains. There's also animals and man in one of these branches. For each species, bacteria, archaea, and eukaryotes, there are viruses, so various are very old. To better understand the different assumptions on the origin of the virus, we have to take a step back and understand how life has started on Earth. Let's remember that our planet formed about 4.5 billion years ago, and water, which is essential for life, started to be present on Earth about 4 billion years ago. The first living entities appeared on Earth 3.8 billion years ago. And we think that this is the case because some fossils were found in Australia, which you can see here in this image, and in these rocks, which date back to 3.5 billion years ago, we found bacteria cells called stromatolites, which can also be found today. On the bottom side of the slide, you can see the stromatolites in the Bahamas, in Australia, and in the Yellowstone National Park. And surprisingly enough, the living beings existing 3.5 billion years ago are also present today. And this is surprising enough. How have they formed? There's not one single answer to this question. The most credible, I would say, assumption is that at the beginning, there were the first constituents of macromolecules, and the first nucleic acids formed. They were made of RNA. RNA has a self-catalytic function, as in they can self-replicate, and so they grew in water over the time, and then they turned into a protein form. This structure then acquired a lipid bilayer, which is the first step of the formation of a cell. So the RNA nucleic acid turned into the DNA nucleic acid. And this is how the protocell came about, leading to life. Now, going back to the phylogenetic tree, there were the bacteria first, then the archaea, and then for endosymbiosis, the eukaryotes. Now, I made this long introduction because it is important to understand that at some point the RNA came about. So, what is the origin of the virus? Now, viruses are present in all cells. Now, there's different models that make us imagine some scenarios. Now, this is the first model, the model of the origin of the virus. There's a system whereby there's a self-replicating RNAs which can get an outer coat or an outer capsid. So this is the first viral structure made up of an RNA genome with a protein shield or a lipid shield. According to this model, virus existed before the cell because the protocell would be nothing but the development of the virus. So the virus becomes more and more complex and evolves into a protocell leading to other cells. So this is the first model on the origin of the virus. So there is the RNA, the virus, and the cell.
The second model is that of viral regression. So first, there is a protocell, and through reductionist evolution, there were portions of the cell that went lost, and then we have a protocell, which is simplified, and it evolved, becoming a parasite. The third model is that of the viral escape. We have a modern cell which and a portion of its cell of genome RNA has acquired the skill to infect different cells, using them as organisms for biosynthesis. Then we have a final model, that is to say the Chimera model, where we have a mix of the other models I already talked about. So we have a pre-cell phase with a diversification of elements starting from RNA. So we will have a protocell with very primitive virus forms, and then we have an evolution of the cell and a co-evolution of the virus. And, this is something that did happen because obviously we can see that in many different examples in terms of eukaryotic cells and bacteria. The cell evolves together with the virus, and the virus evolves together with the cell. So it's a co-evolution. So this is an aspect which is quite certain, whereas we need to understand better the origin of the virus. Also because there is another factor that makes the situation even more complex. In later years, giant viruses were discovered, called, as you see in the slide, giant because they're very big, obviously. You can see here in the picture the dimension of the HIV virus and the bacteria of Escherichia coli, and then we have the virus and the megavirus, where we have dimensions that are similar to bacteria. If we take into account, for instance, HIV, we have about 10 genes. For Escherichia coli, we have about 4,000 genes, whereas this giant virus has thousands of genes. These are very complex structures with the biosynthetic skills. So we have these skills that enables the virus to transfer and to translate and replicate. So this makes the situation even more complex because any assumption on the origin of this virus that has to take into account all these aspects. So therefore, we have organisms that can infect at any level the living beings inhabiting our planet, and they have different forms and sizes. So how do they play a role in the ecosystems? Are they just parasites, or do they have a positive contribution to the ecosystem? Well, since these types of virus are very ancient, we needed to understand why they're still there, if they have a beneficial role to play. So the virusosphere, the world of viruses, is something based on a few pathogenic viruses, or is it something bigger? Well, the answer to this question is that there is a wider diffusion. We have a very, general calculation. In the biosphere, there are 10 times 31 viruses, different viruses. Obviously, this is a very big number, and it's not quite easy for us to understand. Anyway, the stars of the universe, for instance, to give you an idea, are about 10 to 24. So this is to make you see how many different types of viruses there are. And where are they more common? Well, everywhere in the planet. We need to bear in mind that the most part of our planet is made of water, so that means that most viruses are living in water. If we take coastal water, for instance, we have about at least a few million different types of virus for each milliliter of water. The farther we go from the coast, the more the number decreases, and also if we go deeper into the water. But anyway, generally speaking, the number of viruses is 10,000 for each ml, and most of these viruses are bacterial viruses, and they are also present in hot springs, rivers, and lakes, but also in soils, for instance, in arable land or in woodland. But they are present also inside our bodies, and since they are so widespread, they have a role that is important for our ecosystem. And I'll just give you one example. There are so many examples, actually. But anyway, let's bear in mind that viruses are the major predators of the plankton population. So every day, about 20% of the biomass is destroyed by viruses. In particular, we talk about algae and bacterial viruses. So these viruses are very important to maintain the balance of the plankton population, because they guarantee a balanced composition of water. This lysis, because obviously a virus entering a bacteria leads to the death of the bacteria, that is to say that its components are then released, so a lot of organic mass is released, and this is a very important aspect because it makes it possible for other organisms to find organic matter for the rest of their lives. So it is extremely important, for instance, for the camber cycle, the nitrogen cycle, the phosphorus cycle, because these substances that have a non-living, a mineral form are acquired as organic material and made available by viruses to other living organisms. So, well, this is extremely important. For instance, if we think of the balances regarding carbon, the use of the carbon line, the release of oxygen, and so, well, the maintenance of the whole ecosystem. Obviously, this is not the only element, but these viruses are extremely important because they help maintaining the balance of the global ecosystem of our planet. Another important aspect is that viruses play an important role in the evolution of bacteria because they enable a process which is called genic horizontal transfer, with which a bacteria can transfer their genome not only to their progeny but also to other parallel bacteria. So this is extremely important from the evolution point of view for bacteria. So, well, from the ecological, environmental point of view, viruses are extremely important. In order to answer your question, there is another example which I must say it is something I'm really interested in, and that is to say the so-called endogenous retroviruses. I talked about, for instance, the HIV virus. It's a genome, an RNA genome virus that penetrates a cell, and the genome of the virus becomes part of the genome of the host cell. In the case of the hosting cell is a lymphocyte, and this does not enable a transmission of the virus to progeny. It's just a horizontal transmission of the virus. What would happen if a retrovirus would infect a germinal cell? Because in this case, the embryo would have this integrated virus in each cell. Therefore, there would be a process of endogenization that would lead to the fixation of genetic information, because all the cells of this organism, of an animal or a man, would have this virus integrated in a certain part of the organism, like a scar, that is then transmitted to the progeny. So we could wonder whether this is something that really happened. It is a theory, a scientific theory, or it is something that could really happen. Well, actually, this did happen, and it happened many, many times. And it was actually estimated that a significant part of our genome, 8%, has retroviral origins. And to make you understand the importance, the size of this percentage, our proteins come from about 2% only of our genome. So if we say that 8% of our genome has retrovirus origins, well, this is something really interesting. These viruses entered in our genome, in most of the cases, very from the primates, and so we can have a genetic tree of primates and find out in which animal, single animal, individual animal, there was this process of indigenization of viruses, after this infective event, the whole population actually had these specific characteristics down to men, to us, and we do have this starting infective event. Since such a big portion of our genome has been maintained by retroviruses, makes us wonder whether there is a functional role. Well, this is something that is still studied, but we can provide a few examples on how science is developing. A first assumption of the positive role of viruses is the following. They can, if they are integrated in our genome, and this is important because they actually have undergone many mutations, so they started as pathogens, but then in a way, they became inactive, so they do not have any pathogenic function. Each virus, the integrated virus, has its own history. Anyway, these elements are still active, even though they mutated, and they can interact with exogenous viruses and create a defense line, so to speak, because they can, for instance, avoid that a virus interact with a cellular receptor. So we're talking about primates and human beings, but this is true also for other cases. There are, for instance, viruses that integrate in amoeba, and these might be infected by a giant virus, and they can defend themselves by inactivating this virus. A second element, which is extremely interesting, is that they are apparently important also to keep our immune system always on the alert. Many anti-tumoral drugs enable the activation of our immunity defense, and this in a way helps us block the tumor. So the immune system is then activated, and the organism can block the creation, the formation of a tumor. So this is extremely important for this interplay with our immune system. Then there are proofs that show that these viruses are extremely important for the development of embryos in terms of cell polypower, and there are some studies carried out on mice that show that there are some memory processes linked with the expression of these endogenous viruses. We don't know whether this is true for men as well. There is a very interesting example in terms of coevolution between viruses and mammal organisms, is that of the placenta. Here you can see the fetus, and you can see that there is a very specific structure, that is to say, the syncytous trophoblast, that create cells that interact, that make it possible to pass nutrients from the mother to the fetus. So this fusion of different cells is due to a viral protein of the envelope. So we have a viral receptor that interacts with a cellular receptor, and this makes it possible for the virus to enter the cell. Here, the viral protein is no longer something dangerous. It is used by primates and makes it possible to create the placenta. So if there are mutations here, there might be problems in the development of the placenta. And this is something that actually took place in all mammals, studied up to now, because this is a structure that is quite different in the different mammals. But anyway, in all the studies, a viral protein was always involved. So this is a significant example of evolution, co-evolution between an element which is originally hostile, a pathogen, which then is tamed and it is no longer a pathogen and it is used in a beneficial way. So I hope I have explained how a virus is formed, how it replicates, how we can block viruses with the use of drugs and pharmaceuticals, and how our planet actually co-evolved with viruses, and we ourselves co-evolved with viruses. It is a living entity that still has a lot to say, and so we need to study it further and to see how these simple elements can have such a huge impact. Thank you, Professor. Obviously, the fact that these very simple elements actually are so important in terms of life, well, this is actually something astonishing. So for the whole system, in order to go deeper into these themes, I invite you to go and visit the exhibition Living Beings for this special edition of this meeting. You can see it on the website. Thank you. Thank you and goodbye.